sort of keeping up with the discussion and knowing where we're at, and so no, it wasn't me.
Okay.
So was that from CRL or from?
Yeah. I don't remember the name of the girl that is supposed to ship.
Yeah, Rebecca. I think she's
Rebecca.
on the line. Yeah.
Right.
Okay. So.
Hi.
So, hi.
Sorry, I missed the start. I missed the start of that.
No. It's
Just general chat.
Oh.
Yeah.
She was just asking if you were is asking about who's shipping the brains.
Oh, okay. Yes, that would be me. Yep.
Oh, good. So did you have them already?
Yeah, so I just replied to your email. Apologies, I didn't realise that there would be a time limitation on the authorization form. And actually, we haven't collected, I think we were going to send you 3 brains and we don't have all 3, and that's still going to be another couple of weeks. So I did put it in the email to see whether we could extend the date on the authorization form. But we can chat about that in the meeting or offline, whatever works, really.
Oh, okay. Yeah. I would go ahead with the shipment, and we can prepare the paperwork for another shipment if required. I mean, okay, we'll talk about that afterwards.
Yeah.
Okay. Yeah.
Hey, Jose.
Hey, how are you, Ahmed?
Good, thank you. Good seeing you.
Good to see you too. So you survived so far to the snowstorm.
I don't know who is I think it's Pete. That's the snow we have. So
that's my backyard this morning.
Wow.
Yeah.
So you could see it's probably about 50 centimeters or more of snow.
Approximately 50.
So I'm sure you in the UK and Spain are very jealous.
Oh yeah.
We did have a bit of snow earlier in the year, but that's gone now. Well and truly, it's just rain.
Yeah.
Well,
Yes.
I'll tell you something. I spent 2 years in Colorado, in Fort Collins, and I remember that just before Christmas, we had like minus 32 degrees Fahrenheit, Celsius. So really freezing. But you just put on more clothes and that was okay. I came home for Christmas and it was like 4 degrees and raining. I didn't feel my feet warm for the 3 weeks I was in Spain.
Yeah, so we're all digging out here, but we're managing to stay warm. But I think we can proceed with the meeting this morning.
Okay, yeah, Antonio will be joining eventually, but he had some teaching.
Yep, not a problem. So in terms of the agenda, I'll go through a summary of what's gone on since our last meeting. And then because at the last meeting we kind of cut off Jerome and Ahmed, we'll start with Jerome and he'll give us basically an overview of his mouse model. And then, we will introduce Jose. He's a new collaborator from the University of Basque in Spain, and he'll describe some upcoming plans for mass spec imaging of M4K 2009 in brain tissue. And then we'll move on to some of the IND-enabling studies. Ahmed's group has done some bioanalytical method development. We've got feedback from the Tessa Jowell committee. I won't go into that in great detail, but we'll discuss some of their kind of main recommendations and then give an update on the SALT screening results. And then we'll finish off with some next steps. So, since the previous meeting, we do have some new ADME and DMPK data. We did a dog PK study and showed that M4K 2009 has good exposure and bioavailability. It was recommended by Liz at Charles River that we get dog plasma protein binding, and that can help define some of the human dose predictions. So that is in progress. So we should have some results on that in a couple of weeks. We also did UGT phenotyping and inhibition across 7 UGT isoforms. Showed minimal inhibition overall, at least across 6 of them, greater than 20 micromolar. One did have some moderate activity around 10 micromolar, so just something to keep in mind. We also ran CYP induction and CYP phenotyping. There was some signal for CYP3A4 at 10 micromolar, not really at a level that raises a red flag, but it's something to keep in mind. So it was recommended that this was only done at 3 concentrations, that we do a repeat study at 6 concentrations, just to ensure that this is not something that's a big red flag, but if it could be of concern depending on what co-medications patients would be taking. So we do have some results from a high throughput salt screen with M4K 2009. And I've done a few salt screens in the past, and we actually saw multiple crystalline forms across multiple counterions. So we had a fair amount of discussion on which to choose from to scale up. We chose to scale up 2, that being HCl and HBr. But we do have a lot of backups in hand if one of these is not suitable. Both salts existed as both hydrates and anhydrates. So we prioritized the anhydrate for further scale up and characterization. And in that scale up, it showed that the crystal form was reproducible. And both of these have high threshold solubility. So right now we have some accelerated stability studies ongoing. And assuming there's no issues or degradation, and I think we'll be in a good position to pick one of those salts for development moving forward. On the scale-up side, we're just finishing up the review of 4 proposals, and I expect we'll be able to choose a vendor for scale-up of non-GMP material and analytical method development by the end of this week. And then, I mentioned that we did get some feedback from this accelerator program. I think mostly overall, it was very, very positive. They were, I think, particularly impressed by the strength of the team that we've assembled. So I'll touch on some of those recommendations later on in the meeting. So now I'll stop sharing, and Jerome, if you can share your slides.
Absolutely. I'll try to do that. I have a lot of friends in Toronto, and they were asking if now they've officially outdone Montreal in terms of snow. And I was like, I told them it's starting to look like Montreal in October. So I'm not familiar with Teams. I suppose so when it's okay, so I can share specific windows. Suppose that. So can you see my slide now?
Yes.
Yes, awesome, great. Okay, well, it's great to give you a little bit of an overview of what we have in terms of mouse model and what we like to do with the M4K compounds. So in the lab here at McGill, we generated genetically engineered mouse model of that carry the kind of like the hallmark mutations that are found in diffuse midline glioma. So this is what the model looks like. We target mutations to OLIG2 expressing cells with OLIG2 CRE. And then the mutations, of course, it's the histone 3 mutation, the hallmark mutation in the disease, alongside with other lesions. So we're targeting oligodendrocyte progenitor cells here. All these, the mice that develop tumors in this context, so the tumors are spontaneous, meaning that they're not induced by some kind of injection or what. It's not an autotopic model, right? It's tumors that develop spontaneously in the native brain environment. So they're also immunocompetent mice. So we have combined histone 3 with P53, ACVR1, and PI3 kinase pathway activation with the PIK3CA activating mutation, where these models differ in sort of like their of course, genetic characteristics, but also the phenotypes. The model that we think is the best in terms of looking at the effect of ACVR1 so far, based on the data that we have, is the bottom one here, the histone 3 with ACVR1 and PIK3CA. This is what it looks like in terms of survival. So you see the survival curve here. It's a very aggressive model. Most mice will succumb to disease within the first 90 days after birth. What's particularly, I think, good about this model for our purpose is that each of the 3 mutations contribute to the phenotype. So we get tumors if we have histone 3 and PIK3CA, but the phenotype is more aggressive if we add ACVR1 mutation, and we need the histone 3 mutation to be there, and we need PIK3CA mutations to be there as well. So if we think of like basically the potential of testing ACVR1 inhibitors, essentially what this tells us is that this is the kind of benefit that we could expect if we had a perfectly, active drug that would block the ACVR1 completely and kind of make it ACVR1 wild type. So it's probably kind of like sufficient to detect some kind of effect. But because we get tumors with just histone 3 and PIK3CA, this is not a model where we would cure the tumors with just the ACVR1 inhibitor, for example. So this is just something important to keep in mind. Okay. So what we're doing with this is the first thing that we're asking is what are the ACVR1 mutations doing in this model, right? So we know they contribute to the phenotype, but what's happening at the molecular level? And this is going to be useful when we think about looking at the effect of inhibitors, right? So we're comparing mice that have the ACVR1 mutation with mice that don't, and we are profiling the tumors by single-cell RNA sequencing. I'm not going to go into too much details, but just for people that may be interested, our approach is we use a factorization approach. So we use non-negative matrix factorization to basically look at transcriptional programs or gene expression programs that differ between cell states and cell types and between ACVR1 wild type and ACVR1 mutant tumors. To enable us to really get to what are the molecular effects of ACVR1 inhibitors. Sorry, not ACVR1 inhibitors, ACVR1 mutations, and therefore that will guide us in terms of being able to define or measure to what extent ACVR1 inhibitors are effective at preventing those effects of ACVR1 mutations. So what we want to do next, so the analysis of these data, of these data here is underway. We've just collected the first data set. But what we want to do next essentially is take our mouse model and give it M4K2009, and then doing the same type of analysis, right? So the specific question that we're asking here is, to what extent are this ACVR1 inhibitor in particular able to prevent or reverse the effects of mutant ACVR1. We also think we could extend that to a survival analysis with a larger cohort of animals later on. Of course, there's it will require additional planning for that. These are genetically engineered models, so we have we're generating these mice by breeding, and if you do the math, it's basically 1 out of 16 mouse, right, that we get by our standard breeding that will have the combination of all 4 mutations. So we can make that a little bit more efficient, but building a big enough cohort to do a survival study would take some time for sure. So I've talked a little bit, we've talked a little bit before about kind of like, what kind of dosing or schedule we would use, specific technical aspects such as like, what's the vehicle, what's the solvent, what's do these drugs have to be reconstituted every day, every week, and so on and so forth, so that we can plan the logistics of that, and then getting our hands on the drug and then getting the animals and treating them. We would, because of these are, it's an aggressive model, we would probably start treating the mice not too soon, not too long after weaning, I would say, maybe in their 30 to 40 days range, and then maybe give the treatment for 2-3 weeks, and then collect the brains for analysis. That's what we're thinking at this point. That's basically what I have.
Yeah, it makes sense. I mean, when we come to the in vivo part, like when you are ready to do that, we can discuss more about the vehicle and the dosing. I think, like, say, I don't know what other people thinks here, but I think since this is a very aggressive model, I think we can start with the high dose that we know works in vivo, and probably that, and then daily dosing would probably what we need to do.
Of course, there's I mean, I know this drug you've tested it in animals, but in this case, we would be also treating kind of like younger mice. So that's something to think about. Whether they would react differently in terms of toxicity, I don't know.
Yeah. Do you so they are very young. So do you know if you can do the oral dosing on them?
Yeah, so it is possible, but it is, in our experience, it is a bit challenging to do that because we've done oral dosing of other drugs in other mouse model, but around that age, and sometimes it's a bit challenging because of the size of the mice, essentially. But is the idea there to do oral dosing or more like intraperitoneal injection or something
so
else?
This is what I was asking. So we obviously prefer oral dosing if possible, right? So that gives us the more clinical relevance and stuff like that. But if we have to do IP, then we can probably also do IP at a lower dose because it gives you similar kind of exposure with IP if you do a lower, like let's say if you're comparing 100 milligram per kilogram PO and 30 milligram per kilogram IP, for example, just roughly probably give you the similar exposure.
Yeah, so that's when we could potentially. We can think of doing it. We can try, oral dosing. It's worked before. It's just sometimes it's more difficult. For other studies, actually, we're having an approach where we can dose the drug in food, like with the American company that they do that, but your compound has to be amenable to that, and there's also a certain cost to that, and the logistics is a bit more complicated because you have to send the drug or the compound to the US across the border, and they put it in the same mouse diet that we use in our facility, and then we get the food. So it's a kind of simpler and less invasive way, but it's not necessarily amenable for every compound. So yeah, we can think of what's the best, whether we should try IP or first.
If we do oral, I mean, it's a comment for Rebecca. Jerome was asking about the formulation, and what I recall is you make the formulation up weekly and then you store it in the fridge, take it out and warm it up.
Yep. So hey Jerome, nice to see you.
Hi. Rebecca, nice to see you.
Yeah, so we're happy to share all the kind of tolerability data that we have, but as you've mentioned, they're not going to be on mice as young as that you're going to be working with, so it's probably something that you're going to want to look at yourself. I can send you all the information we've got and how old the mice were when we tested them. We're happy to share that. But yeah, as far as the drug, the usual methylcellulose vehicle and a batch every week, dosed 5 days on and then 2 days off at the weekend is what we've been running. But yeah, happy to send you any information if it's helpful. Just not sure if it's going to be comparable.
Done the treatment for in 3 weeks, 4 weeks.
We've actually managed to dose. We extended our license recently, so now we can dose all the way up to 24 weeks. And I think some of our models, as you know, the median survival's quite long, and so it's tolerated well in the age of the mice that we're testing, especially as a single agent. We can send you that as well if that's useful.
Yeah, absolutely. I mean, in our case, of course, we would probably be looking at the more acute treatment because of as I was, I think, showing there, like even if the inhibitor works perfectly, we would still expect the mice to probably succumb by probably 4 or 5 months after birth. Not all the mice without ACVR1 mutations develop tumors, interestingly, at least not within the time span that we've looked at, but most of them will. So that's kind of like the window that we're looking at here.
On the kind of the quicker mouse model that we were using, I think we managed to dose up to the median survival on that's about 40 or 50 days. And so we did manage, I think we started dosing 2 weeks post intracranial injection and then managed to get 4 or 5 weeks out of that. But we kept dosing until they went down rather than dose for set, stop, and then wait for them to come down. So I guess there's a decision to be made whether you continue dosing or not, but maybe with the smaller mice that's less amenable.
Rebecca, have you done any, I guess, IP dosing in efficacy studies with any M4K compounds? I didn't think so.
No, it's all been oral.
Yeah.
Yeah.
So we should probably think of doing it all first, then and just if we encounter any hurdle there, we'll adjust.
Yeah.
I think-
Then we can.
Double check with Deanna whether she's done any of that kind of pre-2020 when she was initially working with it and doing the tolerability. But most recently, the last kind of couple of years, it's all been oral. But I can double check with her.
Yeah, we did a PK via IP a long time ago. That's the only thing we have data for.
Yeah.
We haven't done. I don't know any efficacy study where we dosed IP.
Not that I recall. I think even the early work with Angel was all oral, from what I remember.
And also, one thing I want to mention is that Matthew in my lab was working on this project. He's been also collecting brains to send to Toronto so that they can do some drug binding studies on, I think, with M4K2009, but also other compounds with this particular model.
Yeah. Well, we have compound we're ready to send, but we can certainly discuss offline the best strategy for your model.
So that's where we're at, and we're really looking forward to it. I think it's going to be insightful, and our main goal there really is to get more insights onto the molecular and hopefully on-target effects of M4K2009.
Oh, that would be awesome. Does anyone have any additional questions for Jerome? If not, oh, you have one, Jose? You're on mute.
Yeah. So it's related with our participation in this project. So actually, we're talking here about young mice, right? So pardon my ignorance, but how big is the brain of one of those mice?
I mean, I think the brain. I mean, I don't think the growth of the brain basically doesn't scale up with the growth of the body, like in the sense of like younger mice at the age of weaning. I think they have if I'm not mistaken, a disproportionately large brain compared with their body if you compare it to adults. I might be wrong about that, but that's also my impression just by handling those mice and those brains. So they're not particularly small brains. Also, they do get, because of the tumors, at least at human endpoint, their brains are actually expanded. Like there's maybe a bit of hydrocephalus, but there's so much tumor in there that like you can see on the histology here, right? That it's not uncommon that we open the skull and then the skull is actually bulging, right? That the brain has just become bigger.
Okay, thank you.
Okay, great. So I think now I can turn over to Jose. You have some slides ready?
Yep. There we go. So this is my first meeting with you guys around, well, we had one meeting before this one, but it was really focused. So I wasn't sure about what to show you. So I decided to just give you some context about what we do and what our participation in the project was so far and what I guess that will be in the future. So,
Yeah, that's perfect.
Yeah, so the reason why we are here is because we have some experience in multi-imaging mass spectrometry, especially in the field of lipidomics. Now we combine that technology with other imaging techniques like immunofluorescence or spatial transcriptomics. For example, this image that you see here is just human substantia nigra. You can see only the distribution of two lipids. One that is more abundant in neurons and the other one in white matter, and you can see the whole histology of the tissue, right? So we have some experience in the image of distribution of drugs over tissues, but it was some time ago when actually we did that kind of experiments. Were different experiments. We were trying to incubate the drugs on tissue sections, and they were really difficult because we tried to see the affinity of those drugs for some receptors. So the final concentration of drugs in the tissue sections was really, really low. So we were hitting what I call usually the nanomolar wall, right? That those drugs are usually, when you incubate them, in nanomolar concentration, and that's probably the limit of detection of everything that MALDI can see. So the way in which we do our experiments is we get a biopsy, and a biopsy or a sample, we cryosection it. We cover the section with matrix. This is an organic compound that allow us to extract the molecules that we want to image. Then we define a grid of coordinates over our sample that will become the final pixels in the final image. Introduce the sample in our mass spectrometer, and the mass spectrometer goes coordinate after coordinate, recording the mass spectrum at each of those coordinates. And finally, we use specialized software to reconstruct the distribution of all those molecules that were detected. If you realized, I didn't mention any labeling of any compound. That is good news, and that's also bad news. It's good news because it means that I don't need to label any compound to detect it. So if we see the compound in our multi-target, we can see that in principle the compound in our tissue sections. But in addition, we will be seeing lots of things, mainly lipids in our case, which is again good and bad, because some of those peaks can be so strong that can prevent us from detecting the target molecules. So that's something we have to look to test. But it's also good because in addition to the distribution of the target molecules, we can also learn something from the changes in the metabolism of the tissues after drug administration. Also, we can sometimes follow not only the drug, but also the metabolites of that drug, as we don't need to label them for the detection. So, as I said, the key step in the detection of a metabolite on a tissue section is to have a good limit of detection. That's what we spent our time during these months. We were trying to determine the limit of detection, the LOD, of the two compounds that you sent us, the 2281 and the 2009. This is, for example, the 2281 with two matrices, with and without sodium chloride, trying to help ionizing this molecule. And you can see that we can go all the way down to 10 to the -8 and still have a substantial intensity of the compound. This is very good news, because when you you represent the signal intensity against the concentration, you see that you can go down to more or less 10 to the minus 9, which is fantastic, a fantastic limit of detection. In the case of the compound 2009, we also have the same kind of limit of detection, but there is a difference that I will tell you in a minute. So next steps. I learned about Ahmed told me that you are interested in the distribution, mostly on the distribution of these two compounds in the pons. I learned today that the pons is this part of the brain here. And this is funny because part of the pons is the locus coeruleus, and we just published a paper on the distribution of lipids in locus coeruleus. So this can give you an idea of the best spatial resolution that we can get. These are the distribution of three lipid species. This one is phosphatidylinositol 38,4, and is especially concentrated in the neurons of the locus coeruleus. So these bright spots are individual neurons, or well, mixture of neurons. They are packets of neurons. It's funny because if you add two double bonds to this lipid, instead of being more abundant in the locus coeruleus, you can see you kind of light up the neurons in the mesencephalic nucleus that are just here. So when you represent this lipid, you see these neurons as dark spots. If you represent this lipid, you see these neurons like lighting up, brighten, and you don't see the locus coeruleus. It's a little bit below these neurons. So we have enough spatial resolution to see without any problem these structures here, provided that we have enough signal intensity from the target molecules. So in the meantime, also Antonio, that is also attending this meeting, did all the paperwork to get the clearance from the Spanish ministry to bring the samples from UK. It was faster than we expected. So now everything is ready to reception the first samples. Once we get them, we will get sagittal sections. We define the region of interest, and we will try to determine the best compromise between signal intensity and pixel size. That will determine the spatial structure, the spatial resolution, and what kind of structures we will be able to see in those brains. We will also optimize the procedure for deposit the matrix, maybe by sublimation, that is our favorite way to do it, or by a spraying, or maybe by recrystallization after sublimation, detection of possible metabolites, and so on. So regarding the metabolites, I told you that there was a trick in the detection of 2009. And it's that once you get below 10 to the minus 6, below micromolar concentrations, you don't see the peak from the molecule anymore, but you see other peaks that are related to this molecule that only appear when we add to the matrix this compound, and that allow us to detect the molecule in concentrations of 10 to the minus 8 and below. So we kind of are able to detect this molecule, but not in the expected mass channel, but in other mass channels. And if everything goes well, and we do a good job with the control mice brains, I suppose that at some point you will send us sections or samples of mice with tumors to detect the compounds in those tumors, to see metabolites, I don't know, changes in the lipid profiles, detection of necrotic areas, I don't know, that depends on you. because we only know how to do something, that is do images, and that's it. I mean, I'll be glad to take any questions you may have.
This
I
is
have a,
great. Yeah.
oh, sorry.
No, go ahead. I was just saying this is great, actually. This was very good. This is exactly what we hope to achieve from the experiment, like spatial distribution and what was it showing the resolution. If you can get 10 micrometer resolution, that's great. We know exactly. And the other thing, I think it came out, but it was not obvious, is we can get a concentration effect also in the gradient, right? So Jose, as you can see from your color pictures from your published work, that you can also get an idea of concentration. That's why you're doing the calibration curve. So that's all good, I guess.
I must, yeah, very good points. If we can go to 10 to 10 micron, that I mean, it's really difficult. I don't think that. I mean, that would be a dream, okay? But let's be realistic. Probably if we can go down to 50 micron, it will be great. If we can go to 25 micron per pixel, would be fantastic. If we go down to 10 micron, we can see if the drug accumulates inside the neurons or around the neurons, for example. If it is predominantly in a nucleus or in a different one, yeah, that would be, I don't think that anybody ever published something like that. But this is also the paper I just showed you is the first report on the lipidome of neurons directly from tissue. So this is also the first time. So let's hope for the best. Second thing, multi-imaging is not a quantitative technique. You cannot say, I have this intensity, so I make a calibration curve, and then that means that I have this concentration. Because it may depend on where the compound is. Maybe detecting the drug inside the neuron is different. You have different sensitivity than if it is in the intracellular medium or if it is inside an astrocyte. So maybe we can do a calibration curve just spotting the sample over the tissue sections and try to do some rudimentary calibration curve. We can try to do something like that if it is really relevant. Yeah, but that's difficult to do. These are just relative concentrations.
But
hi, Jose. Yeah, my question was more around the ability to be able to kind of co-localize different things. So you're looking for compound, looking for metabolites, looking for lipids. Will they be on kind of sequential slices, or can you detect all from the same sample slice? Is that correct?
All for the same, but there is all for the same, from the same. We use two polarities, positive polarity and negative polarity. These compounds are amines, so we detect them in positive polarity. The images I saw you are from negative polarity. In positive polarity, you detect some lipid classes. In negative polarity, you detect a different set of lipid classes. We usually run the experiments in negative polarity because we see more lipids. I don't know if you are familiarized with lipids, but we detect phosphatidylserines, phosphatidylethanoamines, phosphatidylglycerol, sphingomyelins, phosphatidylinositol, so a larger collection of lipid families. If you go to positive polarity, you have two problems. One is the problem of the adducts. You detect everything as protonated, sodiated, and potassiated. That's 3 peaks for the same for the same bird, so to speak. So the spectrum the mass spectrum is more complex, and second is dominated by phosphatidylcholine that have at physiological pH they have a positive charge, and they give very strong peaks.
Right.
So yeah, we can in the best situation we will be able to detect that drug, that compound, and maybe 100, 150 lipids that we can identify.
And in terms of kind of a smaller, not lipid based, but smaller metabolites, I'm particularly interested in cholesterol. Is that something that you'd be able to look at in the same kind of
we have detected cholesterol, but that beach always gives a very low signal because it doesn't like to capture either a proton or a sodium or a potassium.
Right.
So it's difficult to detect. We have detected cholesterol in brain samples in a model of brain stroke, scanning at 100 micron per pixel, but we never tried at lower, at better spatial resolutions. So I don't know how deep we can go in the detection of cholesterol.
Right.
We can set the observation window in that region if you're really interested.
Okay,
Yeah.
brilliant. Yeah, thank you.
Thanks, Jose. This looks really, really interesting.
Thank you.
Does anyone have any additional questions for Jose? Okay, Ahmed, I have you on the schedule next.
Yeah, I'll run through it very quickly. Let me see if I can share. Yeah, can you see my slides?
Yes.
Okay, all right. Should I go full screen? See if I can go normally. Okay, is it full screen now?
No, it's not full screen. We are seeing your PowerPoint.
Okay, all right. Okay, so let me just let this always the problem with Microsoft with
Yeah.
Teams. So let me just try and just do it from the regular. Yeah, just give me 1 second. If I can find my cursor now. Okay, okay, I'll just run through from here. So what we did, we are trying to develop a bioanalytical method for 209. I mean, we already use quite a few bioanalytical methods for tissue and plasma in our lab. But what we are trying to do, paying more attention to GLP-like method development, so that when we transfer the method for IND-enabling studies in a GLP lab, they can take this method and have a very good starting point. That's the whole idea of doing this kind of method development, and that's what we were trying to do. And then first thing for this method development is to find so we can do PK and PD studies. And first thing is to find a internal standard. We need to have an IS for this study. Normally for other type of molecule, people use deuterated compound for the IS internal standard. But since we have so many analogs of 209 in our lab, so we thought we are going to find something which we have enough of, and that can be a very close analog, and that can be used as a internal standard. And we found 2163, M4K 2163, and that is what we are using as an internal standard for these studies. Okay, so I'm not going to go over the sample preparation. It's pretty basic sample preparation we do for plasma. So we did the plasma spike and then precipitate the protein and then run it by mass spec. And we did a range of calibration range from 5 to 10,000 nanogram per ml. That's the range we used. And we did QC samples. I'll show you the data. So, and then before I go into that's the mass spec conditions, what we use. I mean, there are many people in this talk or in this meeting are also analyzing the compound, and the mass spec conditions are pretty similar to what we used for our regular or day-to-day analysis of this compound. We are using a gradient. And we are using these Agilent HPLC with a 5500 Q-trap for the analysis. And these are the LC conditions. This is the MS conditions. I just showed you the LC conditions before. And this is how the peaks are looking like. So this is the background, and you can see 10 times more background is our LOQ. So our LOQ is actually 5 nanogram per ml. This is the peak for 5,000 nanogram per ml. Looks pretty good, consistent. And signal to noise, I just said 10 is good. And this is the calibration curve we have. This is 2 run. One run is at the beginning, and then we run bunch of QC samples, and then we ran again. And the average of these 2 run looks very good, very linear. And then when we ran the QC sample, most of them, so there is a 15% difference is allowed for a GLP studies. So most of them came within that 15%, except this one here. But we are just going to repeat that analysis that is ongoing right now. This is we are ongoing. That analysis are ongoing, and we have to do 6 injection from, let's say we take 1 medium concentration, 1 high and low concentration, and then we do 6 injections from there and try to see the reproducibility of these runs. So that is also ongoing right now. We also want to see the short-term stability of the solution. So when we make or prepare the solutions before it goes in mass spec, there may be some delay. So usually we would like to see a 24-hour stability of these calibration solutions over time, and also the solutions from the analysis we are seeing. So these are the short-term stability. We'd just like to see 24-hour stability there. So that is also ongoing. So hopefully we'll finish that, and we can share those to our CRO. And when we had a brief discussion with the CRO already, and they said this is pretty good starting point for them to do the analysis. So that's pretty much all I had to say.
Yeah, I just want to mention that this is really nice that we can do this work internally and not have to outsource this early development work to a CRO. And as Ahmed mentioned, we already spoke with a CRO, potential CRO, and they're really happy with the work that Ahmed's group's done already. So, any questions for Ahmed? Okay. Oh. Yeah, so next on the agenda, I mentioned that we did get the report back from the Tessa Jowell Foundation. So this is their Brain Tumor Research Novel Therapeutics Accelerator. So back in November, Ahmed and I met face-to-face with the committee and presented our work to them. And we just got the report back about the middle of last week. I can share that report with anyone. Just send me an email and I can forward it to you. There's a lot of recommendations in there, and Jose, you'll be interested to know that they were very interested in the mass spec work that we had planned. So that's outlined in that report. But these are some of the, I guess, the main recommendations. There's some other smaller ones within the report, but first and foremost, they kind of wanted more data on potential biomarkers. And this would really be important for if we were to have biomarker driven patient selection. That would really help the trial design. They also want to see some more benchmarking against current standard of care, particularly radiotherapy, and now the recently approved ONC201 in combination studies. And these are things that we'd discussed internally. Actually, a number of the recommendations we'd already kind of discussed within this group, particularly coming back from Charles River as well. But this is something that they really want to see us do is more combination studies. And certainly we've discussed that with Chris Jones' group, as well as Matt Dunn's group in Australia, and now potentially some work with Jerome. So I think it's important to look at particularly the radiotherapy, because Chris has already seen that there's potential for radiosensitization with ALK2 inhibitors of different chemotypes, but this hasn't been done with the M4K compound, so it's something that we should really look into. One thing that we hadn't really discussed internally is what to include in that toxicity study in terms of CNS tox. So this is something that they recommend that we certainly include, because this is obviously a highly blood-brain barrier penetrant compound to assess any risk and benefit with that compound. What was, I think, particularly interesting for me was some feedback from the patient advocacy group, and they suggest that we get a patient advisory board together as soon as possible, really to engage with the regulators early, particularly around our plans for a first-in-human study in children. And this was something that is, endorsed by that committee. They feel that it is something that we should look into, but we'll certainly need a really robust preclinical package to support that. So I thought that was interesting that the patient group is kind of on board with that approach, but they really want to see that we have a lot of evidence to support that is the path forward. So I kind of threw this out to the Connect Consortium to see. I know they already have advisory boards internally, and perhaps we can just move into one of those groups, or do we build that ourselves? And kind of leading into that, although there is support for that first-in-children dosings for a trial, there is concern about dosing at a subtherapeutic dose in children and the ethics around that. So one suggestion is that we do a phase 0 study, a microdose study in adults, to help find an acceptable dose level for a pediatric trial. Like I mentioned, there's a lot of other sort of minor recommendations and risks in that report, but we're going to discuss this, I think, with our steering committee and kind of go through some plans on the recommendation. And certainly I can share those recommendations after that meeting with everyone. But is there any questions or comments on this committee review? Okay. Yeah, so like I said, we're going to discuss this with the steering committee. So with the salt screen, and just to give you some context, I've run programs where we run a high throughput screen and get one crystal form. In this screen, we'd run it in a 96 well plate using 11 counter ions and 6 solvents. And we saw crystal material with 5 of those counter ions. So, a lot of data to kind of sift through in that just initial screen. But we did decide to scale up 2 forms, the HCl and the HBr. And what we found was we find hydrates with each and anhydrate form with each. And what we saw was the anhydrates have actually very high threshold solubility. So we moved forward with those at a higher scale. And we're currently undergoing, like I said, an accelerated stability study at 40 degrees for about a week. We'll look for any degradation in those salts, and then do additional characterization of those salts. And So far, I think everything is looking positive from the data we've seen to this point. No degradation by NMR, which is nice to see. So I think we'll be able to make a decision on one of these salts very soon. Any questions on salt selection? All right. So lastly is just some next steps, and this will be pretty quick. So we received 4 proposals for scale up, and those are being reviewed by OICR. The reviews are finished. We're just kind of going through the evaluation of each of those proposals, and like I said, I think we'll be able to pick a vendor by the end of this week. We are planning to do some additional ADME and DMPK studies. This was recommended by the Charles River team. So we're doing a rat excretion study. So this is a rat biliary duct cannulation study, and we're going to look at levels of M4K2009 in urine, feces, and bile. We could look at metabolites in this study as well. That's quite pricey, so I've decided just to hold off on that for the time being, but we're going to save these samples, and if we do decide to pull the trigger on that, we think it's necessary, then we can go back and analyze those samples for potential metabolites. In terms of clinical planning, that's been a bit challenging to try and get a meeting where everyone is available, but we are trying to follow up on that with the Connect Consortium. But I don't have any update on clinical planning since the last meeting. And that is all I have for today. But I'll open it up to any questions or comments. If not, I guess for next meeting, potentially in a month, if we have some data to go over. If not, probably in March. But yeah, if there's stuff to discuss in a month, then I'll schedule a meeting for this time at the end of February.
Well, it's still warm for Canada. It's now there is no snow, but it's -22. It's going to be on Friday. So
Yeah.
Yeah.
Okay. Finished ahead of schedule this time, so I'll let everyone get on with their day. Thanks very much.
Yeah, thank you.
Cheers.
Thank you.
Thanks.
Thank you.
Bye-bye.
Bye.
Okay. Bye.